In this experiment, bacterial transformation with methods in genetic engineering using biotechnology were used to change the genome of sample E. coli. Genetic engineering allows for genes to be removed, added, or even modified from an organism's genome. Certain bacteria are able to transform their genome by taking in and incorporating exogenous genetic material. This transformation occurs naturally, but can also be achieved in the laboratory. The experiment involved 2 control plates and 2 transformation plates. E. coli cells were treated with calcium chloride and heat, which made the cell walls more permeable. This allowed for the easier intake of the exogenous DNA, which contained the selectable markers IPTG and GFP. These are the results of our experiment. Notice on the control plates, one has growth and the other does not. This is because they both were exposed to the same things and went through the same procedures. But the one with no growth was exposed to an ampicillin antibiotic which kills the cells. Notice the transformation plates both have growth due to the cells taking a plasmid and expressing antibiotic-resistant gene, permitting their survival. As a group, we expected full cell growth of E. coli in favorable conditions of Luria broth medium. However, these cells were exposed to some treatment procedures to alter permeability of their cell membranes. So if all cells recovered during the recovery period, then there should have been full cell growth. As far as our results show, we observed full cell growth. This plate allowed us to make sure that we had viable E. coli cells in a preferable food source. And also it allowed us to see that normal cell growth with transformation should look like. For this plate, we expected no cell growth because it did not receive the plasmid with the ampicillin-resistant gene. Neither did plate 1, but plate 1 was not exposed to ampicillin antibiotic. With no resistance or immunity to the antibiotic, all E. coli cells will die, which is exactly what our observation in the lab showed. This plate allowed us to make sure that the ampicillin resistant was working and allowed us to see what no cell growth would look like. The transformation plates should have been the only ones that contained genetically transformed bacteria cells. These cells were exposed to the plasmid, which contained the selective markers for transformation to occur. We expected cell growth for this plate, but only for those cells that took up the plasmid, transcribed and translated the ampicillin gene, which would allow production of proteins or enzymes that would be resistant to the antibiotic. However, they would not fluoresce because the molecule IPTG was not added. Our observations showed that we did not achieve any cell growth. We expected cell growth as well for this plate, but again did not achieve any cell growth. However, there should have been cell growth of those cells that took up the plasmid. The surviving cells should have fluoresced due to the presence of the IPTG molecule, which attracts a special RNA polymerase to the promoter that will express the GFP gene and result in cells that will fluoresce a green color under UV light. A possible reason why we did not achieve cell growth for plates 3 and 4 could have been because the group may have plated the cells on the wrong side of the agar plate, or perhaps a more likely cause may have been due to the transformation inefficiency of the bacteria cells. Because transformation of bacteria cells is very inefficient, we could have needed thousands of cells on one agar plate to even achieve a minute amount of transformation. So perhaps there were not enough cells that were plated on the agar plate to achieve transformation.